Good morning, genetics students. We're going to pick up where we left off on our overview of the essential vocabulary for the course. When we last visited, we were talking a little bit about the central dogma of biology. And so I just am going to expand a little bit on the idea of the actual bodies that are formed by the DNA, which are known as chromosomes. When we talk about the different cell types in the body, when we hear about a somatic cell is a body cell, so basically any other cell aside from sex cells. And when we talk about the chromosomes that are found inside nucleated cells in the body, there are 22 pairs of autosomes and one pair of sex chromosomes. Females have two X chromosomes and males have one X and a Y. One of the implications about that we'll be talking about at a later date has to do with the presence of a whole body of illnesses known as sex-linked illnesses. A sex-linked gene is typically a gene that is carried on an X chromosome. And because of this, males are at much higher risk for those diseases due to the fact that they only have one X. Females have two X chromosomes, and so the likelihood of having a positive form or positive allele for that particular gene is better. Males would only have to inherit one damaged chromosome in order to express that disease. The other issue has to do with the Y, and the Y is a really interesting chromosome. Of course, the Y is associated with just really a very small set of genes that are specifically contributing to male secondary sex characteristics. So karyotype is something we will expand on. Basically, a karyotype is a picture of chromosomes, and it is typically obtained through the use of a procedure known as amniocentesis, which is done on a pregnant woman. And it would be done for a number of reasons. One of the reasons, obviously, is if there are some markers that show up in her regular blood testing that would indicate higher than normal likelihood that that the fetus that she is carrying has some kind of a genetic condition. In the amniocentesis process, an amount of amniotic fluid is obtained by inserting a needle into the uterus and withdrawing the amniotic fluid from around the developing fetus, and then those cells that are floating around are analyzed, and a relatively simple procedure is done to obtain the chromosomal picture and then analyze the chromosomes, looking for things like, well, a great example, one that we'll talk about, would be Down syndrome. And Down syndrome would have 3 chromosomes rather than 2 at position 21. Or you might have something like a Turner syndrome, where in the sex chromosomes there's only one X and no additional X or no Y. But we'll be talking about those conditions at a later date. So we talked a little bit before about Mendelian traits versus multifactorial traits. And anything, going back to Gregor Mendel, you know that Gregor Mendel was that Austrian monk that did the experiments on pea plants and did all kinds of counting and analysis of his results. And the traits that he studied by far were single gene traits. And from that arose really the entire field of genetics. But what we now know, especially in terms of human genetics, is that the vast majority of the traits that we'll talk about in this course really are multifactorial. They are contributed to by one or more genes, and then there are also environmental factors that influence the expression of those genes as well. This picture just shows a comparison of two traits. That on the left, that's polydactyly, and polydactyly exists in humans as in many other species. You might be familiar with cats that actually have an extra toe, and in the cat world, sometimes it's sort of I guess a desirable anomaly, just kind of making that cat a little bit more unique. But polydactyly is actually a Mendelian trait, and on the right hand side, the slide there is just illustrating the skin color variations, and we'll talk a lot about that because skin color is one of those that there is just an enormous range of possibilities for skin color, and influenced by a large number of um, not only genes but also some environmental conditions as well. All right, so the importance of having an understanding of genes and the risk that those genes might um, have for us, it is very important because it helps us to make decisions regarding our own health. The danger, though, is something that is known as genetic determinism. And genetic determinism is exactly what it sounds like. It's like saying that just because a person has a predisposition for a particular illness, it's an automatic that they're going to have that illness. And of course, if you think about that from the social perspective, um, you realize that that could really be dangerous because it could influence healthcare costs for that individual. It could influence insurance or insurability. Insurability is already a problem. And this is actually why a lot of people do have concerns about genetic databases because they fear that insurance companies will access that information and make decisions about insurability of individuals based only on their genetics and not on how that person behaves and adjust their lifestyle. So a really good example of this would be like a predisposition to heart disease. That doesn't mean that that person necessarily will develop heart disease. It's one of those health questions that as people get older, they'll ask about um, because there are behaviors, diet, exercise, controlling stress that definitely will lower an individual's risk regardless of the fact that that person has the genes that would predispose them towards heart disease. So genetic determinism obviously is something we want to avoid. Making decisions about somebody based on, predetermining that, oh, they have that gene, they'll automatically end up with that illness. Not always the case. When you were in biology, you studied cells pretty extensively. You might remember that all cells except red blood cells, and actually except the sex cells, contain the entire genome. And the red blood cells, we got to qualify that a little bit because actually immature red blood cells do have that. But eventually, as a red blood cell matures, it actually will get rid of that nucleus. It basically sort of spits it out. And the reason for that is because that red blood cell has only one role and it has a limited lifespan, and that role is to carry oxygen. And so a nucleus in a red blood cell really just takes up space that could better be used by filling it up just basically as a sack of hemoglobin and allowing it to expand its function in carrying oxygen. So that's why they don't have a nucleus. They do in their immature state as what's known as a reticulocyte, but as it matures, it gets rid of that nucleus. Differentiation is the process where different lines of cells arise from a certain origin. And what's really interesting about that is the fact that if you think about it, all of the genes for everything are in that original cell that started your life that was known as a zygote. And then now, as a mature human, you literally have thousands of different cell types in your body, all designed for very specific um, function, and and all of that information was in that original cell, but only certain of those genes gets expressed in a certain area of the body. And so that's a pretty fascinating thing. And we talked a little bit about the DNA that doesn't necessarily code for proteins, and those are like those transcription factors, those things that, will turn other genes on and off. We'll talk in depth about stem cells coming up here. It's an area that we're going to take a little divergence on. We're not going to be able to do our stem cell project this fall, sadly, but we will do some substitution activities so that we can better understand the role of stem cells. So this is something you're responsible for, just going back, levels of biological organization, starting at the very simple and moving to the complex. 4 basic tissue types in the human body and their roles. Again, something that you are responsible for. Connective tissues, basically, that word connection is important because things like tendons and ligaments. Tendons connect bones and muscles. Ligaments connect bone to bone. Those are connective tissues. Cartilage, also found in like on the ends of long bones, is another connective tissue. Bone itself is a connective tissue. Blood is actually known as a connective tissue, as is fat. Epithelial tissues are the linings, inside and outside, all those hollow spaces or outer spaces on the body. Muscle tissue, of course, you're familiar with that. These are the cells that provide our ability to move, not only skeletal muscles, but also smooth muscles that move the organs. And then nervous tissue, responsible for transmitting information. Reminder, genotype, that's the alleles that you actually possess. The phenotype is the visible trait or the expression of those genes. A dominant allele is expressed with only one copy. That's all you have to have in order to express a dominant allele, and with a recessive allele, you have to get 2 copies. And that second concept there, the recessives, this is where that term carrier comes from, because a person can definitely carry a recessive that they really aren't even aware that they have. And if they happen to marry and have children with another carrier, that's when you can see that magic combination of 2 of those recessives coming together and that person expressing a recessive condition. We're going to be looking at some pedigrees, and you'll learn how to read a pedigree. Just a couple of things about this diagram here. The squares are the males, the circles are the females. If they're not always in color, obviously, usually you see them shaded in black and white. But if you think about homozygosity and heterozygosity, again, that homozygosity refers to a person having both genes the same for a particular trait, and heterozygosity would mean that you have one of each.